Bonjour à tous. Hello, everyone. So I'm Jacques Fieschi. I'm VP in charge of Research and Development at HalioDx. As you will know, we are a biotech company, and I'm an immunologist by training, so very happy to hear about artificial intelligence. And I asked Emmanuel from my team to join, who is much better than I am to discuss artificial intelligence. So he will present some kind of proof of concept that we've developed in our company. The goal today is to give you a flavor of who we are, what we are doing, some of the issues we may meet in our daily work, and what perspective we have, and where we believe that AI can be helpful for the development of our activities, the company, and our future. So what is HalioDx? HalioDx is a company which is active in the field of immuno-oncology. This is actually pretty new too, as AI maybe it's in the field of informatics. So immuno-oncology is a treatment which was developed for cancer, very effective in some extent in melanoma, non-small cell lung cancer, many other applications. And what we believe is that today and in the next future, there will be a lot of room for evaluation of biomarkers so that the clinician can be better guided into what kind of medication can be given to specific patients. So it goes to new indications, new treatment, and personalized medicine also. So the company is based in Marseille. We are, as of today, 160 people, a bit more in this area. We grow very quickly in the last years, and this growth was also led by a new facility that we opened in the US very recently. We are really science-driven. We have all our activities internally, from R&D to manufacturing, marketing, sales, everything, as much as possible. As you may know, the healthcare field is very regulated, whatever the geographies we are addressing. So this is key to us, and maybe that was a point that was made by the previous speaker about the fairness, the safety, and so on. And this is definitely something that will be critical in the field of healthcare, even if we are looking at diagnostics, because diagnostics is maybe not as regulated as drug, but still pretty regulated. Okay, the company was founded 3 years ago. We have a strong relationship with Inserm and with other companies or institutions, as you may see here. We are organized in 2 business units. One is focusing on diagnostics, and in the field of diagnostic, our flagship product is Immunoscore. So I will show you a few details because that's where AI may be a good option to improve our products. But so I'll go quick on this. The test is, as of today, commercialized for IVD use, so it's CE marked to be used in Europe for diagnosis of colon cancer patients, but also in the US. As I said, we have a laboratory in the US with running tests under CLIA certification. And our other branch is what we call a pharma business unit, where we partner with different industries, pharma industries, biotechs, that may be interested into some tests that we can develop, run for them. So, in summary, what we do at HalioDx is that we develop tests based on our initial product, which is Immunoscore, that we will present very recently. We try to expand our capabilities, but also to have a strong partnership on one side with our customers, which is the pharma industry, but also different groups or companies that can help us to develop our business model. So if we get into more details about Immunoscore, so the idea is to quantify cells into the tumor and at the immediate proximity of the tumor. What is important is that if you may have discussed with pathologists, which are the specialists in the field of the diagnostic that we are looking at, the tissue taken from the patient as a resection or a biopsy is put on a glass slide, eventually stained, immunostained, and analyzed by a trained pathologist. So this is a very interesting field, but the idea here is, for us, is not to replace the pathologist with a machine, even though that could be tempting, but rather to help the pathologist to save time, be more efficient, and eventually provide better results in order to propose a better treatment to the patient in the end by the clinician. To do so, it's critical that we move forward the qualitative analysis and be more quantitative. And we are talking about being quantitative here. We are talking about counting individual cells. This is, after all, manageable. There are already a certain number of tools that are capable to do that. But here we mentioned that we have to look at those cells and count these cells into the tumor, but also at the proximity of the tumor. So we have to define what we call regions of interest and count cells into these different regions. And once we do that, we are able to apply some algorithms that we developed in collaboration with INSERM, and to calculate what we call an immunoscore. This is a pretty simple score, which allows to classify the tumors into different classes that are indicated here. And depending on the class, the prognosis of the patient can vary from poor to pretty good, actually, because here we are measuring immune cells, especially T lymphocytes, into the tumor, and the more of these cells are present into the tumor and at its proximity, the better is the prognosis. And what we believe is that if we would be able to do that, we could, let's say, identify patients that are most likely to respond with a mild chemotherapy, for example, rather than a strong chemotherapy with all the adverse events that we know about this kind of treatment, and that we could make their life, let's say, help them better. On the other hand, we could also identify patients with a low immunoscore, which means a low infiltration of their tumor by T cells, which would benefit for a more stronger treatment than what they would receive today. So this is what was demonstrated in a large international study conducted under the management of the SITC, the Society for Immunotherapy of Cancer, that was published in the Lancet last year, that showed that actually immunoscore works and is predictive of the risk of relapse of the patient with colon cancer. So we would like to bring this technology to an extent where hopefully it can be predictive of the treatment, meaning that we expect that thanks to immunoscore and some other products that are either developed, commercialized, and some others under development today, we believe that we should be able to help the clinicians to better manage the care of the patients, of the individual patients. Looking at not only the tumor, which is still today the current practice, but also look at the immune system of the patient and how does the immune system of an individual react to the tumor and eventually to a treatment, and so that we could adapt the treatment to some specific cases. For example, today for low-grade colon cancer, the standard of care is just surgery, and then the clinician has to decide, should this patient receive chemotherapy or not as an adjuvant treatment. And this decision is not easy because today there is not much to guide this decision. We believe that looking at the immune system and the strength of the response of the immune system of each individual to the tumor will help to decide on should I give chemotherapy or not. Which regimen of chemotherapy should be given. It can vary from, let's say, 3 months to 6 months or more, different compounds, 5-FU, whatever chemotherapy drugs are available. And this will make a strong difference in the life of the patients, either in terms of quality of life, adverse events, but also risk of relapse. So this is why we are there, trying to help the clinicians to take the best care of the patient as they could, and developed the immunoscore. So immunoscore, after all, is it's pretty straightforward, and I wouldn't say low tech, but kind of low tech. And this was a decision to do it this way, because what we want to promote at HalioDx is that our product can be as easily as possible translated into routine. We don't want to develop fancy technologies that will never end into a real lab. I mean, in any hospital or private lab that do routine testing. What we want is to be innovative in a way that we help, at the end of the day, the clinicians for the patients. And for that, we have to remain simple. So by design, immunoscore is a simple technique where we put some kind of innovation, but rather on the interpretation of the data than on the generation of the data. If we look at this slide, we start from on the top from tumor resections, and we immunostain this tissue section for the presence of CD3, CD8 positive cells. I mean, regular T-cell staining, nothing fancy. We use lab equipment that are already present in any regular pathology lab, either at an hospital or in private practice. And after staining, we digitize the slide and do some digital pathology analysis, which is presented here. So I'll be quick so that Emmanuel has a chance to present what we did in the field of artificial intelligence. It's pretty straightforward. There is a short movie on the next slide, so I move to this one. And I try to launch the, can I? It's moving. So what you see here is that there is an area here, I don't see very well, but there is an area here which is actually LC tissue that was determined as tumor by the software as it is today, which means that we need a technician or a scientist to revise the classification of the tissue, because as we said, we need to count the cells into the tumor and in the immediate proximity. So we have to kind of draw a line around the tumor. So we do that today with a software which was trained to do that, but not as smart probably as artificial intelligence. And we spend a lot of time correcting this frontier between the LC tissue and the tumor. And so that's the first field where we believe the artificial intelligence could help us. I'm sure that most of you are convinced of that. But there are many other aspects of our technologies where we think that artificial intelligence could help, like for the detection of the cells and many others. So maybe Emmanuel.
Thank you, Jacques. Maybe I can try that. So first of all, in my team, we are software developers, bioinformaticians, image analysts, and The idea of this project was to enhance the previous machine learning way to do that, which was a random forest technique. And to ramp up the team with that, we involved Euranova, with which we conducted the project I will show you. So the goal of this project is really to minimize the time taken by the experts, because here it's quite a simple example. So for people who are used to look at these images, it's a simple case showed here just for the video, but it can take, it can be a long work to do that. It can take, I don't know, on average more than 20 minutes by image to do that. And for the immunoscore, we need 2 images. So it's a hard work, and it's really a challenge for us to minimize this time doing that. All the other steps are almost automatic or very easy and to achieve. So I'm speaking about this step especially, where we want to train with a new AI technologies. So we are talking about deep learning again. So the idea was, so the method, the pipeline for the training is to use as input the images, the raw data, with the annotations that are done by our experts by segmenting all the image in tissue, normal tissue, epithelium, and tumor regions, which are our 3 regions of interest necessary for giving a good classification. Then it's loaded into the training system, and then the training is done by trying several models, but I think that it converged, it was the UNet that was used almost from the beginning, so the choice was quite quick. So it's a really common model layout used for convolutional neural networks. Then there was a trained models with several parameters. There were dozens of models that were trained using this, and I will show you how. And then the segmentation by patch is given, because we have very big images that needed to be segmented in small patches. And then the reconstruction of the annotation for the whole image was done by post-treatment, and then we have the results. So that is a so-called UNet that was used as a model for doing that. And the metrics for validating each parameters combination and learning cycles where the accuracy, which is a number of correct predictions from all the predictions that were made, and the intersection over union. It was quite more conservative metric, but maybe even more useful for our application, which is the area of overlap between the ground truth and the prediction, divided by the union. And this metric were computed as follows. So the accuracy for all tissue classes, normal tissue, tumor tissue, epithelium. The intersection over union average for all tissues. So there were this done 3 times by images, and then we took the average. Or only for the tumor, which in our application is more relevant, because the score, the immunoscore, is mainly related to the tumor region and the microenvironment. So I'll try to be quick. So the data and the parameters are here. We used 900 pyramidal images. Pyramidal means that they are very large images. So for instance, it could be, this is an average, 50,000 by 40,000 pixels, meaning that in average we have images of 2 billion pixels. So this is also a challenge. And the way they are coded is that they are coded in several layers, so the images are redundant, but it's easier to access. Sometimes, for instance, in this work, we access mainly the level at 5x, 10x, or 2.5x. So the parameters that were, so the images were divided for the training, 80% of these images, 10% for the validation, which is also part of the training process to validate the training, and the tests that were very independent images, 10%. The parameters that were tweaked are the NDPI level, meaning what level of the pyramid should be used, and their context. So we can use as a primary level, one level, and context level, either a context above or a context down. The border probability. So we know that for a good classification in our application, we need to have good classification, especially nearby the border. So we could select the random patches by fixing a minimum number of patches that contains some borders, and also the class weight. We can try, as the tumor is more important for us, to add more weight for the tumor. So I can show you the best combination of parameters with these results, but I will try to go a bit further to the conclusion. So this is the accuracy, so all tissue classes. Again, all the predictions that were good among all the predictions made. We have really good, that was very impressive for us comparing to the previous method. So we have, of course, these outliers, which we always have, but it's easy to say to see. And an important point is that this step for us is always before validation by an expert. So there is a pathologist behind this with a veto rights. So he can say, or he can ask for hand reannotation. So we have the IOU metric on average, which is a bit less, but it's a bit more conservative as a metric, and it's more, I think, relevant for our use. I can show you some examples. So examples of results. On the left is a ground truth. So it was as it was drawn by the expert, pathologist. And on the right side, we have the colors of the prediction with borders in red that correspond to the ground truth. So really, in most of the cases, so in the the big balloon part of the violin plot, we have this kind of results, which are very good because starting from that, we decrease a lot the time to edit this annotation for the experts that have to do this after. So this one is one that we like, and Cécile as well from Euronova, because it looks like a dolphin. And yes, finally, we are talking about these 4 steps. So the importation of the slide is 2 minutes for a human to do it in our system. The automated cells detection and ROI classification by this method, or the previous is 0 minute for the human. And then the manual review of the tumor generation, thanks to the enhancement of the classification, turns from 20 minutes to 8 minutes, times the number of times they do that during the day. It's really helpful, and the validation is still 3 minutes. So the conclusion is that for us, the tissue classification is a key performance in a production setting. So it doesn't enhance the immunoscore, because at the end it's always the expert pathologist who had the final word, but it's in in production settings it's crucial. Yes, of course, the improved tissue classification, automatic classification reduces the need for manual adjustment, obviously. And also we have some perspective around the AI for other other applications, mainly within the immunoscore. For instance, to detect automatically the artifacts. We have in this kind of image a lot of bubbles, some hair, some wrinkles that could be spotted automatically with this system. And yes, to extend it on other indications than colon. Thank you for your attention.